Hello, this is Jim Schnabel with the 14th lecture in a series from Principles of Biochemistry, Albert Lehninger's 7th edition. Today we'll be discussing carbohydrates, polysaccharides, and review the nomenclature and basic chemistry, and lay out an introductory talk on this subject. So carbohydrates are the most abundant biomolecules on Earth. They are probably the closest intersect between biochemistry and organic chemistry. There's a lot of overlap between those particular areas. They have multiple functions. They function as energy. They have structural components to them. They're involved in lubrication of joints, also recognition molecules and adhesion molecules as well. The empiric formula is CH2O, ergo the name. There are mono, di, and polysaccharides. Polysaccharides generally have 20 or more units to them. Monosaccharides can be aldehydes or ketones. They have 2 or more carboxyl groups. And you can get reaction with alcohols to form either ketals or acetals. There are alpha and beta forms where we talk about the attachment either at the front or the back of the number 1 or number 2 carbon. And the most useful projection that we use is Haworth perspective formulas. So when we look at Haworth perspectives, we're using a planar representation of the molecule, but we have a bold front to this, which indicates that this is jutting out of the plane of the surface. This is going to the back of the surface, and then we have the orientation of constituent groups up, and these are on the same side, and the constituents going down or on the opposite side. We have in here an internal ester linkage here, and we number the carbons, and you can have either a we can either have a 6-member ring or 5-member ring, and so we talk about 6-member rings as pyranoses and furanoses, and these are some examples. So we talk about alpha position, which is the orientation of the hydroxyl group. We can also talk about beta, where the hydroxyl group is above the plane, and the alpha, for example, the alpha fructofuranose, where the hydroxyl group is below, and beta, where it's above. So this is a furan skeletal moiety here, 5 rings, 5 constituents in the ring, and pure pyran, where you've got 6 with double bonds present in these or in this orientation here, although these are pyranoses here, so these are hydroxyl groups that are attached. So again, the groups listed on the right pointing down, and then you've got a chair configuration. So one other way of doing this is to orient the front of the molecule going out of the plane of the surface and the back of the surface with the axis to the center, and the chirality of the molecule is dependent on what the mirror images would look like at this point. So 2 possible chair forms for beta D glucopyranose, for example, are present here, and you can have orientation around this particular axis here and be able to visualize the relative location of the oxygen and the moieties that are present, whether they're folded up or folded down. They reverse their inverted forms of each other in this configuration here. This is another example here with the central axis, and again the orientation of the ring around this axis here. So we talk about different constituent groups attached. They can be amines, they can be oxidative forms, reducing forms, deoxy forms, forms that are involved in acidification. The metabolic intermediates are phosphorylated generally. This is an energy intermediate. The phosphorylation will trap the carbohydrate in cells and facilitate subsequent chemical transformation. You can also have disaccharides, which are two of these linked together. There's a glycosidic bond, which forms an acetal ring link from a hemiacetal and alcohol, and this condensation eliminates water from the reaction. So here's an example here, formation of a disaccharide. So you get elimination of the water, the proton here from beta D glucose and alpha D glucose. Again, the difference between the alpha form is the presence of the hydroxyl group, whether it's below the plane of the paper, it's C1. Again, this is a orientation from 1, 2, 3, 4, and 5. 6 with the methylene group attached to the hydroxyl. You can have a linear form of this, which we'll be showing shortly, but there's elimination of water, condensation with elimination of water, or hydrolysis, where you add water, break the bond, and you get the two constituent groups. But at any rate, the oxygen being down will form here, and that'll be eliminated, and the orientation is such that the oxygen will be bonded in the alpha position of this. And this is maltose, which is a disaccharide of two glucoses. Both of these are alpha glucose and beta glucose. And in an open chain structure, they can also reduce copper. The nomenclature, again, you've got configuration of the carbon, alpha beta, the carbon 1 joining the first monosaccharide. The first one is by definition on the left. You name the non-reducing residue, so you have a furano, so the configuration, whether it's alpha or beta, and then you name the non-reducing residue in the second part, and then after that you either have furano, which is a 5-membered ring, or parano, which is a 6-membered ring, and in parentheses you can indicate the carbon atoms are joined by the glycosidic bond that was formed through the elimination of water, and then you name the second residue. So in order, it's configuration, non-reducing residue, either furano or parano, then in parentheses, whatever the carbon atoms are that are involved with the glycosidic bond, and then the second residue. So an example of this, which the shorthand version is lactose beta form, but the official chemical name would be beta D galactopyranocele, and then the connection is from 1 carbon to 4 carbon, 1 carbon of the galactose and the 4 carbon of the glucose. That's the non-reducing residue. The reducing residues comes next, glucopyranose, and then the transition. This is another way of doing it, galactose beta 1 to 4 glucose. So there are 3 ways of giving this. This is the chemical name, and this is the most informative. The other ones would require some element of sense that galactose is galactose plus glucose. Sucrose, which is your common table sugar, same principle. Beta D fructopyranocele, alpha D glucopyranocide. So this is the structure of it. The other words, you could call it sucrose, just like you do here with lactose, or you could just say fructose 2 beta alpha 1 glucose, which is the same as glucose alpha 1 2 beta fructose. That would simply be the reverse of these two, but this is a principle that's involved in each. Polysaccharides are in most cases a storage form. So you have starch in plants, glycogen in animals. These are breakdown products for energy. They're storage forms for energy. They can also form structural elements. So you have cellulose in plants, bacterial membranes. Additionally, there's no defined molecular weight. They really have no molecular weight. Starch consists of 2 glucose polymers, amylose and amylopectin, so there's branching involved. The amylose is connected by alpha 1,4 linkage, and the amylopectin is highly branched. This starch is a difference from glycogen, which has alpha 1,4 linkage and alpha 1,6 branches of glucose. Is more highly branched and is compact than is amylopectin. Cellulose is a beta-1,4 glucose polymer. Animals cannot metabolize cellulose unless you have a symbiotic organism, such as termites or ruminant animals have the ability to break down cellulose into its component glucose molecules. Wood is cellulose with other polymers such as lignins. 30% of the wood mass consists of that entity. Chitin is N-acetylglucosamine with beta-1,4 linkage, and the C2 has an acetylated amino group. When you have a polysaccharide structure, you have the same issues as with proteins. You can have a tightly coiled helix. Hydroxyl groups can point out. Cellulose, each monomer orients at 180 degrees to its neighbor, so you have maximum hydrogen bonding to cause high tensile strength. This is the good property of wood. Heteropolysaccharides can be a polymer of two different residues in bacterial cell wall. Lysozymes hydrolyze a glycosidic bond, and you also have carbohydrates comprising the extracellular matrix, which is a gel-like material. It facilitates the fusion of molecules. It's comprised of glycosaminoglycans, which are not found in plants. It's formed by two types of monosaccharides. One of these is N-acetylglucosamine, and a uronic acid, which is either D-glucuronic acid or L-iduronic acid. So this is nomenclature that you can absorb as, but again, unless you have the structures, you really cannot address that. But they're just strictly points or tenets of that make these particular polysaccharides particularly special. These polysaccharides are negatively charged, and negative charges are located on alternate sides. If you took the ring form, the paranos or the furanos, and you hydrolyzed to eliminate the ester bond, you'd have a linear form. Hyaluron is a synovial joint fluid lubricant, highly viscous, found also in the vitreous humor of the eye. You can also have some other polysaccharides such as chondroitin, dermatan, keratan, and heparan sulfates. These are in connective tissue. They contribute tensile strength and elasticity. The entity of Ehlers-Danlos syndrome is a autosomal recessive inherited condition which has genetic defects and weaknesses of the connective tissue. Wound healing is a problem. Glucoconjugates include proteoglycans, glycoproteins, and phosphosphingolipids. These are information carriers and also recognition sites. They are involved in membrane attachment and recognition. Proteoglycans are sulfated glycosaminoglycan binds the membrane. These are integral to membranes and they can be shed into the extracellular matrix. These are involved in recognition and signaling functions, and they serve as models for thrombosis and coagulation. Glycoproteins are found on the outer face of the plasma membrane and of various organelles, as a Golgi complex, for example, or secretory granules or lysosomes involved in degradation. Glycosphingolipids are found primarily in neurons. They are used for nerve conduction and myelin formation. Proteoglycan aggregates are extremely large assemblies of proteins and hyaluronan, which form connective tissue and form a fibrous matrix. Glycoproteins are smaller than proteoglycans. They have a variable carbohydrate content. There's a hydroxyl, either serine or threonine linked. They comprise secreted products such as mucin, immunoglobulins, smaller hormones such as follicular stimulating hormone, luteinizing hormone, and thyroid stimulating hormone. Glycolipids and lipopolysaccharides are membrane components. Gangliosides are a component of neural membranes. Lipopolysaccharides comprise the outer membrane of gram-negative bacteria, and they can form endotoxins when released. We talk about carbohydrates as informational molecules, and a smaller number of conformations exist than what you would otherwise predict from the structural pathway alone. Synthesizing enzymes possess conformational specificity, so some carbohydrates will work with an enzyme only if it has a certain conformation around the carbon chiral center. Lectins are proteins which regulate function. They have bound oligosaccharides. Luteinizing hormone and thyroid stimulating hormone are examples. The oligosaccharide binding confers protection from hepatic uptake and destruction. These hormones are under rigorous control. Lectin recognition also facilitates leukocyte migration to initiate and extend an immune response. Viral sialidase is an influenza enzyme which removes the host cell membranes and it releases viral particles for reinfection and spread. This is another polysaccharide. Bacterial lectins, Helicobacter pylori lectin binds to gastric epithelium, especially blood type O, and causes gastritis and possible non-Hodgkin's malignant lymphoma. Intermediate forms have a lymphocyte increase or proliferation, which predates the lymphoma. That's a different stage of this process. And blood parasites also use lectins as part of their pathologic process. Carbohydrates possess recognition sequences which facilitate reactions. This is a type of code. And the lectin provides the same analog as an enzyme binding site. And then finally, for the chemical assessment of carbohydrates, these are linear polymers. They can be characterized chemically. For heterosaccharides, you can use enzymes to remove the lipid or the protein moiety. And for the lab techniques, again, the overlap with organic chemistry is pretty clear here. You can use column, gas, or solid chromatography, mass spectroscopy, and nuclear magnetic resonance study. And you can utilize sequential removal of monomers, and you can do microarrays with fluorescent labeling to further characterize the carbohydrate under consideration. That's it for this lecture, and we'll be moving next to nucleic acids. Thank you very much.